Hello everyone, my name is Dr. Priyanka Pradhan. Today I am going to start a video lecture on basics of carbohydrate chemistry, later on glycosylation. Carbohydrates are the body's primary and most efficient source of energy, which is essential for brain function, muscle activity and overall survival. It contains carbon, hydrogen, oxygen molecules. The general formula of carbohydrates is CnH2O whole N. Plants use sunlight to convert carbon dioxide and water to glucose and oxygen, which is the fundamental life-supporting process on Earth. One can define carbohydrates as polyhydroxyaldehydes or ketones or molecules that yield them upon hydrolysis. Naturally occurring sugars are D sugars, which is determined by the orientation of hydroxyl group on the carbonyl carbon, which is furthest from the carbonyl group. Here this OH is the right side, this is called D sugar, and here this OH is the left side, this is called L sugar. Carbohydrates are classified into three major groups. First one is the monosaccharides. This is the simplest version of carbohydrates. It contains only one sugar group. It cannot be hydrolyzed further, such as glucose, fructose, ribose, glyceraldehyde. Next one is the disaccharides. When two monosaccharides are linked via glycosidic bond, then this is called disaccharides, such as sucrose, lactose, maltose. And the last one is the polysaccharides. When more than sugar units are combined to each other, then this is called polysaccharide. This is examples include starch, glycogen, cellulose, chitin. Chemists represent carbohydrates in two projection system. First one is the Fischer projection. This is the linear representation of monosaccharides. It shows the open chain stereochemistry of carbohydrates, so one can distinguish between D and L configuration. Next one is the Haworth projection. It represents the cyclic ring forms and anomeric orientation. When this C1 aldehyde reacts with the C5 hydroxyl group, it gives alpha and beta configuration. Here this is the alpha because OH group is the below, here this is beta because OH group is the above. So in aqueous solution, glucose interconverts between these alpha and beta D-glucopyranosides via this open chain structure. So this is called mutarotation. This is the sugar moiety. Here lots of chiral center are there. Based on this we can classify it as glucose, galactose, mannose, rhamnose like this. And glucose and galactose are C4 epimer. They are differ only in the C4 position. Glucose and mannose are C2 epimer. The saccharide units are covalently bonded via this anomeric position, and we can call it as this is the glycosides. There are three types of glycosides are there. Here C3 group is not there, so this is the ferrier glycosides, and this is the 2-oxy, and this is the 2-deoxyglycosides. And the synthesis of this 2-deoxyglycoside is very challenging because the absence of C2 directing group here. And these glycosides is very much important because a wide range of natural products, drug molecules contains this glycoside as a chiral building block. And we can protect this hydroxyl group using various protecting groups such as silyl, benzyl, methyl, benzoyl, acetyl. And based on this reactivity, we can define it as armed or disarmed donors. So this for glycosylation reaction we need donor, acceptor and promoter, and this glycosylation reaction goes via this oxocarbonium ion intermediate. If the reaction goes via SN1 pathway, it will give mixture of stereoisomer alpha and beta. If it will give if it will go via this SN1 SN2 pathway, it will give alpha, either alpha or beta configuration. But in between this alpha and beta configuration, this alpha configuration is more stable one due to this anomeric effect. And the stereoselectivity of the reaction are influenced by various factors such as presence of neighboring group participation, anomeric effect, solvent effect, presence of leaving group, protecting group, and indirect approach, presence of stirring hindrance, nature of glycosyl donor and acceptor, etc. Thank you everyone.